Well, welcome back to day 2 on week 5 of the semester. Today, we are going to start discussions about chapter 13. Technically, chapter 13, because we are talking about carbohydrates, is the first time that we are introduced to a biomolecule. So chapter 13, since we are talking about carbohydrates, technically, this is our first chapter in biochemistry. So understand that right now we are going to learn about molecules that contain hydroxyl groups, and that's why we talk about them right after chapter 12. But carbohydrates, it is an exciting chapter. It's one of my favorite topics to talk about. This is what I did my PhD in, studying carbohydrates in cancer cells. And I proceeded to do studies in my postdoctoral studies in Canada, understanding how carbohydrates are utilized by yeast in order to, do different products like fermentation of cocoa beans to make chocolate, or even for fermentation in order to produce beer. So carbohydrates is something that it is very useful, and we all need them, even if you try to avoid them. So let's get started talking about carbohydrates. When it comes to carbohydrates, understand that this is a major source of energy from your diet. It is important to note that it is actually the preferred energy. Is one of the main sources. Many biochemical pathways put in energy in order to make carbohydrates, in order for us to derive energy from these type of foods. Carbohydrates can be found anywhere, and you may be aware of some of the sources of carbohydrates there. In the image that we have in the right side of the slide, you can see pasta, potatoes, you can see rice, you can see beans. So carbohydrates are in many, many food products. Now, carbohydrates, when it comes to chemical composition, it is composed of carbon, hydrogen, and oxygen, okay? Now, sometimes we don't hear the word carbohydrates. Sometimes we hear the called saccharides, or we also hear the word sugar, okay? Now, in the bottom, we see a chemical reaction, okay? And in the forward direction, we see that when CO2, H2O, and energy is actually coming together through the process of photosynthesis, and understand that photosynthesis happens in plants. Nature is synthesizing a carbohydrate, which is called glucose. Glucose, as we're going to see later in the chapter, is a monosaccharide. Now, in the process of respiration, okay, which is one of the things that we do in humans, okay? In the process of respiration, we can take that glucose molecule, combine it with O2, and produce CO2, water, and energy. In terms of the energy that it is produced by the process of respiration, this energy is chemical energy. And we discussed energy several weeks ago, but just to remind you, this chemical energy is the energy that is stored in bonds, okay? And the major molecule in our bodies that is going to be in charge of this chemical energy is called ATP, adenosine triphosphate. That's what it stands for. And when this molecule specifically undergoes the process of hydrolysis, basically it means that it's going to be, the bonds are going to be broken utilizing water as one of the reactants, then that chemical energy, it is put into the system. Now, let me ask you guys, just so we are reminding ourselves that this reaction is actually not foreign to us. what is the classification of the respiration reaction? So remember, we classified these reactions as combination, single replacement, double replacement, decomposition. Excellent, exactly. So just imagine, this is why I think organic chemistry and biochemistry is such an amazing field, because even when we look inside our bodies and how these molecules actually enter in the reactions that our body actually can do, it is amazing. As you can see, and it was put in the chat, a combustion is what happens every time we actually consume, foods, and our body combusts those biomolecules to make CO2, water, and energy. You may be like, but wait, where does that CO2 go? Every time you breathe, you guys, especially when you're breathing out, that's the CO2 that is made in the body. That water is going to go into the water molecules that we have. That energy, it is utilized for all the biochemical pathways that need to be done in order to maintain the human body. And sometimes we think of combustion, it's like, oh, it's this big explosions and you can see them in action movies and all that kind of stuff. No, the body actually does a combustion in a controlled system. Now, in this class, we do not go over the process of metabolism, okay? But understand that this combustion reaction, which actually is tied in with oxidation and reduction reactions, releases energy little by little to provide everything we need in order to carry all of the processes that the body needs to undergo. Now, let's go back and deal with carbohydrates, and let's look at the classes of carbohydrates that we will be studying in this chapter. So when it comes to the types of carbohydrates that we are going to be dealing with here, even though there's more types of carbohydrates, we are mainly going to be focusing on monosaccharides. A monosaccharide, it is the simplest carbohydrate because we're looking at one unit, okay, of a carbohydrate molecule. And you can see in the bottom of the slides that we have the monosaccharides, glucose, fructose, and galactose, which is among the most common ones that we have out there. But understand that these are not the only monosaccharides out there. We are also going to talk about disaccharides. And the prefix di actually tells us how many units of monosaccharides we have in these molecules, which is 2. The disaccharides that we are going to be focusing on is going to be sucrose. Sucrose is what you may know as table sugar. Then we have lactose. Lactose is milk sugar. Maltose is as what we know as malt sugar. Okay, so again, all of these are disaccharides. Lastly, we have polysaccharides. And that prefix poly means many, okay? Polysaccharides are actually polymers of many monosaccharides. Now, in this course, when it comes to polysaccharides, we are only going to be focusing on polysaccharides that are composed of glucose molecules. And to give you the example of one of them, this is a molecule for starch. We're going to see how starch is actually made out of 2 polymers out there, but this is just illustrating, okay, a polysaccharide, which we know as starch. Now, when it comes to classification, we can actually further classify our monosaccharides, and that is going to be done by two things. Number one, the number of carbons that we have in our monosaccharide. Again, we're talking about 1 unit of sugar. And depending on the number of carbons, which you can see that is going to be from 3 to 8, we can classify our monosaccharide. We can also classify our monosaccharide depending on the carbonyl functionality that we have. So when it comes to the classification based on the number of carbons, you can see here the different terms that we're going to utilize to classify those monosaccharides. So if our carbohydrate, okay, our monosaccharide has 3 carbons, it's called a triose. If it has 4 carbons, it's going to be tetrose. 5 carbon atoms, pentose. 6 carbon atom, hexose. The 7 carbon atom is going to have Greek prefixes, also the 8 carbon atoms. So if we have 7 carbon atoms, in my monosaccharide, what would be the term that we utilize for 7 carbon atoms? If we use excellent heptose. What about 8 carbon atoms? Great job. Octose. Now, when it comes to the carbonyl functionality, if your monosaccharide has an aldehyde group, then that is known as an aldose. You can see here the molecule of erythrose. It is an aldose because it has an aldehyde in its structure. We can also have monosaccharides that have a ketone group. And to be honest with you guys, in all my training in sugar chemistry, I have never seen a monosaccharide that has both the aldehyde and the ketone in it. At least in nature, we haven't found one that I'm aware of. But if your monosaccharide has a ketone group, then it's known as a ketose. Okay? Now, understand that we can incorporate these two terminologies, okay, into one word. So when we look at the examples that we have here, we have glucose and fructose, which are 2 monosaccharides. Now, we know that since it has an aldehyde, it is an aldose sugar. So that's why we have a LDO in front of hexose. The hexose is just representing that glucose has 1, 2, 3, 4, 5, 6 carbon atoms. That's where the hexose comes from. In the case of fructose, understand that fructose, when we look at this, it has a ketone in its structure. That's why we see keto, okay, in front of the word that we utilize for the number of carbons. And similarly, that hexose represents that we have 1, 2, 3, 4, 5, 6 carbon atoms in our structure, okay? So this word, aldohexose or ketohexose, is representing both classifications together. So at this time, let's practice classifying monosaccharides. So I will do the first one with you guys, and then we're going to move on to the other two. So when we look at this first image, we're going to count the number of carbons in my molecule. There's 1, 2, 3, 4, 5. That means that this is a pentose sugar. Notice that the majority of the names for carbohydrates end in OSE, okay? Glucose, fructose, galactose, okay? And that is true for many monosaccharides. Now, when I look for the carbonyl functionality here, we can see that this has a ketone. So the classification for this monosaccharide is going to be a keto pentose. So what makes this a ketone is going to be that carbon that is double bonded with oxygen, and then that is attached to carbon atoms on either side. The reason why we see it as a T shape is because this is called a Fissure projection. It is a way to illustrate monosaccharides. It is actually discussed later in the chapter. So what I highlighted in gray, that's what makes it a ketone. Let's move on to the next illustration of a monosaccharide. Take a moment. Let's classify in terms of the carbonyl functionality and the number of carbons. Let's put it in the chat. What is the classification of this monosaccharide? I agree with the heptose. Let's put the number of carbons. 1, 2, 3, 4, 5, 6, 7. So we know it is a heptose. So is it an aldo or a keto? So I see aldo and keto. Excellent job. I also see this is an aldose sugar. Here, to complete the octet, there's a hydrogen. So this is an aldo. Heptose. Okay, let's move on to the next illustration. Now, illustrated, we only see about 2 carbon atoms, but understand that this is called a Fissure projection. Every time we have lines crossing, understand that there are carbon atoms. So there's a carbon atom right here, and there's a carbon atom right here. So based on that, what is the classification of the following monosaccharide? Great, that's a tetrose. I agree with that. Aldo or keto? Great job. This is an aldo, tetrose. Tetrose because we have 1, 2, 3, 4 carbon atoms. Remember, yesterday in class we learned that CHO is the condensed structure for an aldehyde, because this is the same thing as having C double bond O, H. Questions on how to classify monosaccharides, depending on the number of carbons and carbonyl functionality. Then let's move on to the next subject, which is the concept of isomers. We learned in chapter 11 that we can have a molecule that has the same chemical formula as another one. So if you guys recall, we were looking at the case of C4H10, which means that this molecule is going to have 4 carbons and 10 hydrogens. And we saw there how we can have a molecule that is just a straight chain alkane like butane, but we could also see a molecule that has a substituent. So we observed how butane and 2-methylpropane had the same chemical formula, C4H10, but they had different structures. Understand that there is a term that is utilized whenever we have molecules that share the same chemical formula, but they have different arrangement of atoms. And at times, since they have the different arrangement of atoms, then they're going to have different properties. Overall, that is known as an isomer. And when it comes to isomers, we're going to learn right now the different types of isomers that are out there. Again, this is just a limited view. Isomers is a complex subject in organic chemistry. And then if you're taking organic chemistry for a whole semester or the whole year, understand that you spend at least a month understanding isomers precisely, just because of the complexity of the subject. But here, I'm just giving you an introduction of what isomers are and the different types of isomers that we have out there. So in this flow chart, we have the different classes of isomers that we are going to learn in this course, starting with constitutional isomers, okay? So what makes a constitutional isomer? Let me just define it in the corner. A constitutional isomer are molecules, and you need to always compare two molecules that have the same chemical formula but different arrangement of atoms. So here, the atoms are not going to be connected in the same spot. So when we look at, for example, what we have here in the two examples, we are comparing the molecule on the top and the molecule on the bottom, okay? So the molecule on the top is going to be 2-fluoropropane. The molecule on the bottom is 1-fluoropropane. When we look at the chemical formulas for both of them, okay, they are going to be C3H7F. If you look at the molecule at the bottom, we also have that C3H7F. So again, I'm comparing two molecules. They're going to have the same chemical formula. If I name them, they're going to have different names because the attachment of the atoms is different, okay? And that's what we know as constitutional or structural isomers. In chemistry, we can also have stereoisomers, okay? And let's define what a stereoisomer is. Again, we're going to compare two molecules. When we're comparing molecules, and you're always comparing two, remember, that have the same chemical formula, and the only thing that they differ, that are different from each other, is in the spatial arrangement of their atoms. Now, understand that overall, when it comes to stereoisomers, we're going to have two different flavors. First are going to be diastereomers. And when it comes to diastereomers, again, even though there's a larger class of compounds that are called diastereomers, and diastereomers pretty much means that you have two chiral centers. We're going to talk about chiral centers in a moment. And then they're going to be mirror images of each other. But in this course, every time you see cis and trans isomer, and you know that they're cis and trans isomers, they're going to be labeled diastereomers. So for this course, you guys don't need to, confuse yourself with like, how do I know if this is diastereomers? When you see that the pair of molecules given, one is cis and the other one is trans, okay? And they have the same name, you're going to label them diastereomers. The other class of stereoisomers that we have are enantiomers, okay? And when it comes to enantiomers, we can see here at the pair that we have a carbon atom that it has 4 different groups attached to it, okay? This is what is known as a chiral molecule. We will next going to talk about chiral molecules. Now, understand that when it comes to this, the molecule that we see on the right is the mirror image of the molecule on the left, okay? And that's what makes them be enantiomers of each other. These are going to be 2 molecules. We can see that they share the same chemical formula. They can even share the same attachment of the atoms. It's just that one is the mirror image of the other, okay? And that is what we consider a chiral carbon. So in general, when it comes to isomers, you guys need to understand how to distinguish between constitutional or structural isomers, diastereomers, and enantiomers. And after I finish the discussion talking about chirality, we will have some practice problems. So let's dive in and talk about chiral molecules, okay? So the term chiral refers to anything that cannot be superimposed on its own mirror image, okay? So understand that not all, but certain organic molecules are chiral, meaning that they are non-superimposable mirror images, okay? Now, this concept of chirality is very important because biology lives in a chiral world. So understand that this chirality is how sometimes the body actually looks at the molecules that are incoming, okay? So for example, we even have chiral objects around us. So I'm just going to turn on my camera. So when it comes to this, you guys, we have our hands, okay? And if we think about it, our hands are going to be mirror images of each other. So you see that the thumbs are reflecting each other, our index fingers, our middle fingers, our ring fingers, our pinkies are reflecting each other. Now, these two hands are chiral. That's why we have a right and a left side in our bodies. Now, if I try to superimpose the image, which means that I'm going to take my hands and put them on top of the other, as you can see, they do not superimpose. And superimposing means that they're going to be lining up. The same thing happens with our feet. Our feet are non-superimposable. That's why when we buy shoes, there is a right shoe and there's a left shoe, okay? So there's many objects around us that are going to be chiral, okay? Now, on the other hand, socks are not chiral, even though they go on chiral objects like our feet. Shoes are chiral because those objects cannot be superimposed on one another, okay? Now, what does it mean in terms of chemistry, this whole concept of chirality? Well, we need to know that when it comes to this, the main thing is that when we look at a carbon atom, your carbon atom, in order to be chiral, let me write it on the notes. A chiral, and please, this is not pronounced chiral, it's chiral. I want to specify that. Remember, you don't study chemistry, you study chemistry. So the right way to pronounce it is chiral. So a chiral carbon atom must be bonded to 4 different groups. And that's what makes a chiral carbon, 4 different things. That's why you see that the carbon atom here is attached to that black ball, that blue ball, that magenta ball, that red ball, okay? That means that molecule A is chiral. What is molecule B? Molecule B, it's its mirror image, okay? So what happens is that if you have a chiral carbon and you have its mirror image, okay? When you try now to take that first compound and you try to flip it to try to align the atoms, we can see in the image on the right that molecule A and molecule B are non-superimposable, meaning that they cannot line up like our hands. So they are not the same molecule. So when you have 2 molecules, they're each chiral, they're mirror images of each other, and they are not superimposed. That means that they are enantiomers of each other. Okay? So this concept of enantiomers, you guys, comes from the idea that in the laboratory, okay, when these molecules are made, scientists started noticing that they will get pairs of compounds like this. And then sometimes they try to separate them. Sometimes they're hard to separate. But now they had to distinguish of like, wait, but then these 2 things that have same chemical formula, same attachment of the atoms, but they're arranged differently in space. Sometimes they even have different properties. So we need to distinguish them and we need to assign them a name, and the name is called enantiomers, okay? Now, understand that not all molecules, when you look at their mirror image, they're going to be non-superimposable, because we also have molecules that are not chiral. We have objects that are not chiral, okay? So when you have an achiral, or in other words, not chiral molecules, when you try to look at the mirror image, okay, of the first molecule, they are superimposable. That means that they lined up. And because they can be superimposed, they are what they called, or what we called, same structures or identical molecules. So understand that if you start with a molecule that is not chiral and you look at its mirror image, because both images can overlay on each other, meaning superimpose, we're just talking about identical molecules. Let me illustrate to you guys an achiral or not chiral object. So in a laboratory, as you can see, we have this example of a flask. This is an Erlenmeyer Flask. It is an instrument that is utilized to measure volume. So if you have two flasks, okay, and there's no markings on the flask, basically, if you try to overlay them, okay, they will be able to overlay, in other words, superimpose, okay, because the flasks are not chiral objects. Similarly, as I just mentioned, if you start with a molecule that is not chiral, the reason why this carbon is not chiral is because it has two groups that are the same. So if you have two groups that are the same, three groups that are the same, four groups that are the same, you're not a chiral carbon. Now, in the instance in which you have a carbon atom that is not chiral, and you compare it to its mirror image, when you rotate them, you notice that these two molecules are going to perfectly overlay on each other. So a molecule that is not chiral, along with its mirror image, they are basically identical structures, okay? So first, I want to go over a problem in which you guys are given pairs of molecules. And we need to determine if these pairs of compounds are either identical, they are diastereomers, they are enantiomers, or they are constitutional isomers, okay? So let's work on these 5 problems together. So we solidify this concept, okay? So we can see here that this is, again, a Fissure projection. In a Fissure projection, understand that in the intercept, there is a carbon atom. So when we look at this, the carbon in the middle is chiral because it has 4 different groups around it. It has the CHO, it has the hydrogen, it has the bromine, and it has the hydroxy or hydroxyl group, okay? So when we look at this, the hydrogens are pointing at each other, the bromines are in the back, and then we see the CHOs and the OH. These 2 molecules are mirror images of each other. So what will be the classification of this pair of molecules where they are same chemical formula, they're mirror images of each other? Great job. These are enantiomers of each other. Let's move on to the next pair. When we look at these 2 molecules, let me write in the carbons and hydrogens for anybody that will benefit from that. This is a CH3, this is a C, this is a CH, this is a CH3, this is a CH3, this is a carbon, this is a CH, this is a CH3. The molecule on the left, let's practice for this week's exam. What is the name for the molecule on the left? Let's put it in the chat. Name for molecule on the left. Excellent. This is 2-fluoro-2-butene. What about the molecule on the right? It's not 1-fluoro. Yeah, it has the same name. This is also 2-fluoro-2-butene. Now, when we look at the molecules, again, these are alkenes. Is our fluorine in the same environment in both molecules? If we look at the molecule carefully on the left, you can see that the fluorine in the CH3 is pointing in the same direction, right? What I mean by this is that this and this are pointing in the same direction. When you look at the molecule on the right, this is not pointing to the CH3, it's technically pointing to the hydrogen. Okay. Let's not do constitutional yet. Excellent. Yes, this is cis and trans. So which one is cis and which one is trans? So what about the first molecule? Is that cis or trans? Correct. This is cis. This is trans. So when we have two molecules and the only difference between the two is that one is cis and one is trans, as it was written in the chat, they're going to be diastereomers of each other. Okay, so remember, for the purposes of this course, whenever you see two molecules, same name, okay, it's just that one is cis and one is trans. Diastereomers is your classification. Let's move on to the next pair. Here we have two molecules, okay? When we look at the molecule on the left, it has 1, 2, 3, 4, 5, 6 carbons. So this is C6. And then it has 5, 8, 9, 14. It has 14 hydrogens. Okay. When I look at the molecule on the right, one, two, three, 4, 5, 6, C6, H14. Okay. Take a moment and name them, the molecule on the left and the molecule on the right. And let's put it in the chat. What is the name of the molecule on the left? No, that's not the correct name. No, that's not the correct name. What is the longest carbon chain on the molecule on the left? How many carbons? 5. Excellent. That is 3-methyl-pentane. What about the molecule on the right? What is the name? Exactly the same name. 3-methyl-pentane. So what is the relationship between these two molecules? They have the same chemical formula, same name. Exactly, thank you. This is 2 identical molecules. There's going to be 2 instances in which we have identical molecules. This, which is same chemical formula, same name, or if you are given 2 molecules that there's no chiral center and they're not superimposable. And you will know that you're dealing with something like that because it's going to be given to you in a Fissure projection. Let's move on to the next example. Okay. So the molecule on the left, you guys should know what it is. What is the molecule on the left? We talked about it in chapter 11. What's the name of that? Excellent, that's benzene. The chemical formula for benzene, when it's just a benzene ring, is C6H6. Okay? Remember, each of the vertex has a carbon and they have a hydrogen. Let's look at the molecule on the right. Let me add in the carbons and hydrogens here so people see it. CH, 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 CH. Let's look at the molecule on the right. This is a CH2. This is a CH2. This is a CH. This is a carbon. This is a carbon. This is a CH. What is the chemical formula of the molecule on the right? Excellent. C6H6. 2 molecules, same chemical formula, but obviously not the same attachment of atoms. What is the relationship between them? Look at how you can categorize them. Identical, diastereomers, enantiomers, or constitutional isomers. Exactly, these are constitutional isomers. Remember that the concept of achiral It's just letting you know that there's a carbon atom that doesn't have any chirality, meaning it doesn't have 4 different groups. What we're categorizing here is if the pair of molecules given is identical, diastereomers, enantiomers, or constitutional isomers. Okay. Let's look at the last one. I'm going to give you guys 2 minutes to categorize these 2 alkenes. I'll give you guys 2 minutes to categorize these 2 alkenes. Let's get started. First of all, we have to establish if these 2 molecules have the same chemical formula. Let me write in the number of the carbons and hydrogens. So this is a CH3, this is a CH3, this is a carbon, this is a CH, this is a CH2, this is a CH3. So the molecule that I have on the left has 1, 2, 3, 4, 5, 6 carbons. C6. It has 3, 7, 8, 13. No, 12. And 12 hydrogens. Okay. Let me look at the molecule. On the right, this is a CH3. This is a CH. This is a C. This is a CH3. This is a CH2. This is a CH3. Let's look at the molecule on the right. We have 1, 2, 3, 4, 5, 6, C6. And then we have 4, 7 plus 5, 12 hydrogens, okay? So in these molecules, let me go ahead and name them. The molecule on the left, this is 2-methyl 2-pentene. When I look at the molecule on the right, this is 3-methyl 2-pentene. We have 2 molecules, same chemical formula, different names, so that means different attachment of atoms. What is the relationship between these 2 molecules? Excellent. These are constitutional isomers. You have to go through the same process. What I don't want you guys to do is that you're working too fast in the exam, and then you look at an alkene and you're like, oh, alkenes are always diastereomers. No, because in order, as we saw in the second example, for having molecules that are diastereomers, they need to have the same name, and the only difference between them is going to be that one is cis and the other one is trans. So again, the take-home message from this, if you have something that is identical, like the example that we saw on the first row, you're going to have same chemical formula, same name. That makes them identical. Diastereomers, you must have same chemical formula, same name. The only difference between the names is going to be cis versus trans. If you have enantiomers, you're going to have a pair of molecules in which you have a chiral carbon, and they're going to be mirror images of each other. Constitutional isomers, we're going to have the same chemical formula, different attachment of the atoms, like we can see in the first example in the bottom row, or same chemical formula, but different names, like we saw in the second example in the bottom row. Any questions on how to identify a pair of compounds as identical, diastereomers, enantiomers, or constitutional isomers? Go ahead with your question. No problem. Let's now solidify the concept of how to identify chiral carbons in an organic compound. Okay, so here we have two molecules, okay? Remember, the concept of having a chiral carbon means that the carbon atom has 4 different things, okay? It must have 4 distinct groups around them, okay? So let's deal with the molecule on the left first. Let me write in the carbon atoms. This is a CH3. This is a CH2. This is a CH. This is a CH2. This is a CH3. So if I'm trying to determine the chirality of something, understand that I need to treat every carbon and understand, do my carbons have 4 different things around them? Or are they attached to 4 different things? Let me label the carbons with numbers. This is carbon 1, 2, 3, 4, 5. Okay, so carbon number 1 has 3 things that are the same. So that means that carbon number 1 is not chiral because it's attached to 3 hydrogens. Carbon number 2 is attached to 2 things that are the same. Remember, I need 4 that are different. So because it has 2 that are the same, then this carbon is not chiral. Carbon number 3 is attached to hydrogen, to bromine, to the left side of the molecule, which is a CH2CH3, the right side of the molecule, which is also a CH2CH3. So that means that carbon number 3 is also not chiral. What about carbon number 4? Is it not chiral or chiral? So NC or C? What about carbon number 4? Is it chiral or not chiral? Look at what it's attached to. It's attached to a CH3. It's attached to the rest of the molecule and 2 hydrogens. Exactly, it has 2 hydrogens. So this carbon is not chiral. What about CH3s? Is a CH3 a chiral carbon? So you can see that this molecule, in other words, 3-bromopentane, does not have a single chiral carbon. So if I need to classify overall this molecule is not chiral because it doesn't have a single carbon atom that is chiral. Let's look at the molecule on the right. 2-bromopentane. Let me add in the hydrogens and the carbons. This is a CH3. This is a CH2. This is a CH2. This is a CH. This is a CH3. I'm going to number the carbon atoms. 1, 2, 3, 4, 5. Okay? So, as we just learned, carbon number 1 is a CH3, is not chiral. Carbon number 3, 4, 5, they are all not chiral. So again, 1, 3, 4, and 5, they're not chiral carbons because they have 2 or more groups that are the same. In this case, hydrogen. Let's look at carbon 2. Carbon 2 is bonded to hydrogen, bonded to bromine, bonded to this area of the molecule, and bonded to all of this. Carbon number 2 has 4 different groups around that carbon atom. So that means that carbon number 2 is a chiral carbon. So if your molecule has at least 1 chiral carbon, then the molecule overall will be considered chiral. So when it comes to chirality, if you find that your molecule has at least 1 chiral center, that makes the whole molecule chiral. If it has no chiral carbons, the molecule is not chiral. Let's practice. Here we have the following molecule. This is 2-methyl-hexane. Let me add in the carbons and hydrogens. This molecule has, and again, this numbering has nothing to do with, let me actually do it in color because I don't want people to confuse that with the numbering that I do whenever we're naming. So this molecule has 7 carbons. Take a moment, look at all of the carbon atoms here. Let's put in the chat the letter for the carbon in this molecule that is chiral. Excellent answer, none. A, C, and G are not chiral. They have 3 hydrogens. D, E, and F are not chiral. They have 2 hydrogens. Then that leaves us with B. The reason why B is not chiral is because we have a hydrogen, a CH3, a CH3. Oh, wait, 2 groups are the same immediately. Letter B is not chiral. So carbon B is not chiral. So when I need to classify if this molecule is chiral or not chiral, then this molecule overall is not chiral. Put a 1 in the chat if you're ready to do these problems on your own. Put a 2 in the chat if I still need to work through this one before I let you guys practice. Excellent. So let's just put in the carbon atoms here. Let me label them with letters. Again, these letters are at random, you guys. This is A, B, C, D, E, F. Take a moment, okay? And cross in your brain which ones, based on what we have discussed so far, are not chiral. Let's take a moment. And let's cross out whatever based on the definition. We know it's not chiral. So through this process, you guys should have in your brain, you know what? A is not chiral. B is not chiral. D is not chiral. E is not chiral, and F is not chiral. Put a 1 in the chat if it makes sense why all of those letters I crossed out, because they are not chiral. For those of you that are like, Dr. Luz, I don't see what you see. Remember, if you have 2 atoms that are the same, then immediately that carbon is not chiral. So A, D, and E have 2 hydrogens. They cannot be chiral. B has 3 hydrogens. It cannot be chiral. So understand that as long as you have 2 or 3 that are the same in terms of atoms, then all of a sudden you're not chiral. Now let's look at C, okay? And let's look at the situation of C. C is bonded to hydrogen on one side, CH3 on one side, CH2 on one side, CH2 on the other side. At first you will say like, oh, CH2 and CH2, those are the same. This is not chiral. But hold on a minute, because whenever you're looking at this, especially if you're in a cyclic system, you have to look, and we already did this exercise before. We have to look beyond. We did this for cis and trans. You have to look at beyond that first CH2 to see if indeed they are the same or whatnot. And we also did this when we were looking at reactions before. So when we look overall at this area of the CH2, versus this area of the CH2, are they the same? Yes or no? They're not the same, right? So then that means that hydrogen is a distinct group. CH3 is a distinct group. That whole area where the top CH2 is one different group, that area in the bottom that I label it violet is a different group. So carbon C is chiral. Whenever we have a chiral center in a molecule, we identify it with an asterisk. So this is a chiral center. So overall, the molecule is chiral. When you have a ring system, that's a great question. When you have a great ring system, you have to at least look at one beyond the first set of atoms that you attach. So that's why we're going past the CH2. So you look a little bit, you look to the next atom down the road. Especially in cyclic system, if you have a straight chain alkane, please look past the first atom to determine if indeed they're different groups or not. Okay, let's move on to the next example. You guys got this. I'm going to write in the carbons and hydrogens. Okay. Ooh, somebody has it already on the chat. Excellent job. I hope that everybody can identify it. So again, you're writing the carbons and hydrogens. And there are some that should be crossed immediately. F is not chiral. E is not chiral. D is not chiral immediately because they are CH2s and CH3s. Why is A and B not chiral also? Let's put it in the chat. Why is A and B not chiral? Not necessarily because it is a CH2. That is also a thing. Because remember that in order to have a chiral carbon, you need to have 4 different things. The carbons in an alkene, okay, we cannot say that they are chiral carbons because they don't have 4 different things attached to it, okay? That leaves us to explore letter C. And as you can see, in letter C, we are attached to a hydrogen, we are attached to this area of the molecule. That carbon atom is attached to this area of the molecule. And it is attached to this area of the molecule. So carbon C is our chiral center. So this molecule overall is chiral. Let's do one more example. A and B are not chiral because they are trigonal planar. Remember that by definition, you need to have 4 different things attached to a carbon atom. If you're trigonal planar, you're only attached to 3 things. So by definition, we don't say, oh, this is chiral. Just because we're attached to 3 things instead of 4. So that just say that that is not. Let me write in the carbons and hydrogens. It's a CH, CH2, CH2, CH, CH, CH3, CH3. What will be your reasoning behind this molecule being chiral? Look at the molecule closely. Let me label these guys but with letters. Let's put in the chat. What are the letters that, based on all the examples that we have done, for sure are not chiral carbons? Let's put them in the chat. Which letters are not chiral? Excellent. G is not, it's a CH2. B is not, it's a CH2. F is not, D is not. Okay. Now, that leaves us with E and with A, E, and C. Let's start at the top. Is A chiral? Is A a chiral carbon or not? Is A a chiral? Great observation, you guys. It can't. Let's look at A. It has a hydrogen. Excellent. It has an OH. Awesome. But when we look at this path, and when we look at this path, they look the same. Look, even down to the substituents. If I take the route on the left, it's a CH2 connected to a CH, and that CH is connected to a CH3. If I look at the path on the right, okay, CH2 bonded to CH, that's even bonded to a CH3. So A is also not chiral. Let's now look at E and C. Let's look at the path for C. It's bonded to hydrogen. is bonded to a CH3. Is bonded to a CH2. And is bonded to a CH. Can C be chiral? Excellent. So for this molecule, this is a chiral center. What about E? Excellent. So this molecule has 2 chiral centers. So the molecule will be chiral. Questions trying to determine if a molecule that I'm working with is chiral or not chiral, in trying to define what are the chiral centers that are present in it. One more thing about chirality. It is important in biological systems, okay? Let me give you guys at least 2 examples so we can see how chirality can actually give things different properties. Here we have the structure of limonene. Limonene is a chemical compound that is present in the peels of citrus fruits and is also present in plants. Limonene is utilized in foods, beverages, and even chewing gum because of the flavor that it has. Now, we in this class don't identify our pair of enantiomers based on R configuration or S configuration, but that's how basically, as you can see, this topic is more extensive, but that's how we can actually categorize enantiomers and give them a label. And we call them R or S configurations, depending on the direction of where the groups are at in terms of space, okay? Now, if we look at limonene, in this molecule overall, you guys, there is only 1 chiral center, only 1, right here. 1 chiral center in the whole molecule. The one on the left, we call the R configuration. I'm not going to explain that in detail. You guys don't need to label things as R or S. The one on the right is called S configuration. The simple fact that limonene has these 2 enantiomers, they give different properties. The R configuration on a limonene is what gives you the flavoring of orange. The S configuration in limonene is what gives you the taste of lemons, 1 chiral center. So these 2 enantiomers have completely different properties based on 1 carbon that is putting one of its group spatially different, okay? So this is very interesting. Just that, imagine just having the position in terms of space of 1 group give these distinct properties for the molecule, because it's going to have the same chemical formula, it's going to have the same attachment of the atoms. It's just that in space, the dashed line means that that whole group that you see with the CH2, so that, you know, group that kind of looks like isopropyl, but it has a double bond in it, that's facing the back. The one on the right is facing the front. That's why we put it on a wedge. And that gives complete different properties. One tastes like orange, and then the other one tastes like lemons. Where do we see chirality also? In something that maybe some of you guys use in everyday life. That is medicine, okay? Here I have the molecule of naproxen, okay? So maybe at some point in your life, you needed to take naproxen or your doctor gave you naproxen, okay? This is an anti-inflammatory medicine. Now, in this molecule, similar to what we saw in limonene, there is one chiral center, you guys, only one, this carbon. So when naproxen was synthesized, both enantiomers were produced, okay? But the interesting thing, not all enantiomers have this, but some do. Enantiomers, we saw in the case of limonenes that can trigger different properties. They can even trigger at times completely different physiological responses. So whenever you're ingesting naproxen, just because your doctor gave you an anti-inflammatory, you have muscle pain, so on and so forth, okay? Naproxen is Aleve, commercially known as Aleve. Let me double check, but I think it is Aleve. Naproxen. Yeah, naproxen is Aleve. So if you take Aleve, you have taken naproxen. Now, the one that you're ingesting, you guys, is S-naproxen. Because it is known that that is the naproxen that it causes anti-inflammatory properties. Now, this naproxen, when it was synthesized in the laboratory, needed to be separated from its enantiomer. Because in the laboratory, it was discovered that naproxen, in its R version, it actually causes toxicity in the liver. So in a case in which when you go in the laboratory, you synthesize these drugs, okay? This is why clinical trials in laboratory work is so extensive when medicine is put out there. It takes years for these things to be available in the market because they need to understand that when this is synthesized, if there's two enantiomers, what are the real effects on the body? And as you can see, the R configuration of naproxen causes toxicity in the liver. So this drug specifically, they need to separate the enantiomers, and what you ingest in Aleve, which is the commercial name for naproxen, is only the S configuration one. Now, understand that that is not true for every single medicine or drug that is out there. Most drugs that are on the market are sold as mixtures of enantiomers. Because for some cases, not the case of naproxen, but in some cases, they have seen that the pairs of enantiomers, one is the one that works and the other one doesn't cause effect. So then, you know, the company that is synthesizing it, they'll be like, you know what, we'll leave both enantiomers in there. Now, why is it that they may cause different effects? Because whenever we have chiral molecules entering in the body, they need to be received chemically by what we call in biochemistry receptors. And these receptors, as you can see, have, and this is just an analogy of, I don't know if you guys have played that game Operation in which you have shapes that you're putting them in the hole. Just think about that in your cells, you're going to have these biomolecules that has specific spaces for a particular configuration of the pieces being together for a molecule. So they need to fit the correct receptor in order for these molecules to actually show, you know, and do relieving of whatever they have to do biochemically in the body. And the body cannot take any enantiomer. Sometimes it's going to take a specific one. This is why it's so important that we understand the concept of enantiomers, because as you can see, it has a lot of effects in biological systems. Any other questions when it comes to isomers? No problem. So the wedge means that the bond is facing us, it's poking to the front. The dash means that that bond is facing the back of the plane. That is just a way that we illustrate it. So wedge means pointing towards us. It's like pointing towards the screen. The dash means that it's like in the back, like towards the back of the plane. Since there are no other questions, right now we're going on break.